Hello everyone, welcome to molecular biology. Our topic is about recombination in sexually and asexually reproducing organisms. Before we proceed to our main discussion, these are the learning outcomes that you students should have after this lecture. So first, you must understand the process of recombination in sexually and asexually reproducing organisms. Second, learn concept of Mendelian laws in pattern of inheritance. And third, differentiate sexual and asexual recombination. Fourth, understand the process of variation of genes through recombination in sexual reproducing organisms. And lastly, understand conjugation, transduction, and transformation of asexual reproducing organisms. These are the terms that we are going to encounter all throughout this discussion. So recombination, it is the rearrangement of genetic material, while phenotype and genotype don't get confused among these two because phenotype it is the observable characteristic of an individual, while genotype it is the individual's collection of genes. Duplex, it is the double-stranded polynucleotide molecule. Let's proceed to the first part of our lecture. So, ever wonder why you look different from your sibling even though you both have same parents? As you can see here in the picture, you can see that the other offspring inherited his mother's hair, while the other his father's. So, in this lecture, we're gonna learn this variation. Recombination is the mixture and assembly of new genetic combination. It occurs through the process of crossing over or physical exchange between molecules. Recombinant is the molecule or an organism that holds a new genetic material. Sexual recombination happens in meiosis. To be specific, in prophase 1. Duplicated chromosomes will line up and recombine to each other by crossing over and will eventually get each other's genes, and this will result into recombinant chromosomes. So these recombinant chromosomes will be randomly assorted into gametes. So these gametes already contain the chromosome of each parent. Finally, the gametes will merge, one from the mother and one from the father, forming a zygote. And this resulting offspring will contain a new set of genes from both parents. The nature of this recombination is also manifested in the combination of inherited traits of the subsequent generation. That's why you might have your grandfather's nose or your grandmother's nose, depending on which trait is dominant during your generation. The way in which traits are passed on from one generation to the next, and sometimes skip generation, was first explained by Gregor Mendel. Gregor Mendel, he discovered that genes come in pairs, one from each parent, and are inherited as distinct units through his work on pea plants. In his experiment, he used two types of peas, the round gray and wrinkled white, wherein after the gamete production and fertilization, it only produced all round and gray. And after the self-pollinization and fertilization, all the possible traits are visible. So, based on Mendel's laws, each generation of sexually reproducing organism is a new combination of parental genomes. Dominant, this is the expressed trait, usually seen right after the fertilization. As you can see here in the picture, he used the yellow and green pea as an illustration of his experiment, and it only produced the yellow peas. On the other hand, the recessive trait, wherein he used the yellow peas, but this time it produced the green pea. So recessive, it is the hidden trait. Punnett square. It is used to predict the genotypes of a particular cross or breeding. Genotypes, as I said a while ago, these are the individuals' collections of genes. So this Punnett square can be used to determine the probability of an offspring having a particular genotype. So mixing of genes generates genetic diversity, increasing the opportunity for more robust and well-adapted offspring. Recombinant DNA technology. It is used to control the mixing of genes rather than relying only on the natural mixing of the whole genomes. Single genes can be altered, replaced, deleted, or moved into new genomes. The ability to manipulate single traits has implications not only in the laboratory, but also potentially in the treatment and prevention of diseases. Let us now discuss the processes that are involved in the recombination in asexual reproduction. Recombination in asexual reproduction can happen in three ways. It could be in conjugation, transduction, or transformation. Conjugation. It is the process by which one bacterium transfers genetic material to another through direct contact. There are two types of bacteria that participate in this process, the F-positive and F-negative bacteria. In order for this process to be successful, the two mating bacteria should be in contact with each other, forming a filamentous bridge. J. Lederberg and William Hayes demonstrated the polarity in conjugation, wherein F-positive bacteria can transfer its genetic material to F-negative, making the F-negative bacteria into F-positive and infecting another F-negative bacteria. However, F-negative bacteria cannot transfer its genetic material to the F-positive because F-positive bacteria has the fertility factor, wherein the F-negative bacteria doesn't have fertility factor. The fertility factor is responsible for the establishing of physical connection between the two mating bacteria. The fertility factor or the F factor is an extra chromosomal circle of double-stranded DNA or plasmid carrying the genes coding for the construction of the mating bridge. So as you can see here in the picture, this is the chromosomal DNA and the plasmid which are responsible for the building of this connecting bridge or the filamentous bridge. And then, after the construction, the F-positive bacteria will transfer its genetic material to the F-negative, as you can see here. And after that, the old donor will remain F-positive. And then here, the F-negative bacteria a while ago became F-positive also, capable of transferring its genetic material to another bacteria. Fertility factor can also insert itself into the host chromosome through a crossover or recombination event. This high-frequency recombination bacteria can pull host chromosomal information into the recipient bacteria, and another recombination can insert that information into the recipient chromosome. This is how the high-frequency bacterial chromosome infects another F-negative cell. First, it will transfer its genetic material into the F-negative cell, and then the transferred strand is replicated, as you can see here. And crossing over takes place between the donated high-frequency chromosome and the original chromosome of the F-negative cell. So just like animal and plant viruses that infect their eukaryotic cells, bacteria, which is a type of prokaryote, also has viruses that infects them. So transduction, it is the process by which a virus transfers its genetic material from one bacterium to another. In early 1960s, Francois Jacob and Ellie Wallman studied the transmission of units of hereditary carried by virus from one bacterium to another. So these viruses that infect bacteria are called bacteriophages. And an example of a bacteriophage is the bacteriophage T4. It is a specialized protein coats that enable viruses to insert their DNA through the cell wall of the bacterial cell. Just like what you see in this picture, bacteriophage can infect E. coli and use the host to synthesize new phage particles. Lytic cycle. This is the process in which a virus synthesizes itself inside the bacteria. So step 1, the attachment, wherein the phage attaches to the bacterial cell and the genetic component of the phage enters the bacterial cells. Following infection, the viral genetic component dominates the cellular machinery of the host cells and leads to viral reproduction. Subsequently, new phages are constructed and the bacterial cell is lysed. In 1952, Alfred Hershey and Martha Chase, they confirmed that the DNA of the bacterial virus was the carrier of its genetic determination in the transduction process. So, Hershey and Chase used T2 bacteriophage in their experiment to identify whether DNA or protein is the genetic material, and to define the function of the protein coat and nucleic acid in the viral reproduction process, they radioactively labeled the viral protein and viral DNA. So they used sulfur 35 to label viral protein and phosphate 32 to label viral DNA. So finally, the last part of the asexual reproduction, the transformation. So it transfers DNA from one organism to another without the protection of conjugative bridge or viral coat, unlike the first two that requires the conjugative bridge or a viral coat. So in an attempt to develop vaccine against pneumonia, Frederick Griffith made an experiment wherein he used a virulent and not virulent type of Streptococcus pneumoniae. He first to identify the bacterial transformation in which the form and function of a bacteria changes. As you can see here in the picture, the smooth type bacteria has a protective layer wherein it inhibits the phagocytic cells of the host in engulfing them. That's why they can multiply and can cause pneumonia. So, in Frederick Griffith's experiment, he killed the smooth cells and then he injected it in the mouse, and the mouse remains healthy and no colonies were isolated. The next thing he did is he added these heat-killed S cells to the living RAF cells or the R cells and injected it in the mouse. But the mouse contracts pneumonia and eventually died. And there were also rough and smooth colonies that are isolated from the tissue. So after Frederick Griffith experiment on transformation, there were scientists that are also curious about his experiments. That's why Oswald D. Avery, Colin McLeod, and M.J. McCarthy identified that DNA is the transforming material. This is what they did. So they used enzymes, the protease, ribonuclease, and DNase. So the protease and ribonuclease treatment did not stop the transformation. However, the DNase, the deoxyribonuclease, it stopped the transformation, or it prevented the transformation. So they concluded that DNA is the transforming material. That's all for today. Thank you for listening.